Okay, good evening, everybody, and thank you so much for being here tonight. This is a Conversations and Cocktails, I think Conversations, Food and Cocktails, about a disease, multiple myeloma, also referred to as one of the plasma cell cancers or dyscrasias, and I'm joined by four myeloma experts here. I'm going to give some introductory remarks, and then each one of them is going to speak for a little bit. Dr. Landgren will introduce the program, the institute, and then I get to drill them and ask some questions. I think if you haven't already provided us with your question, please do so, but also we'll have time, I hope, at the end for people to raise their hand and ask questions. So I have been fortunate enough to be the director of the Sylvester Comprehensive Cancer Center for nearly 13 years. I came to Florida from New York City with my wife. I had been at Sloan Kettering for 20 years, and the goal here in South Florida is and remains to build a destination cancer center to attract the best and brightest in the field, to build teams around some of the top physicians and researchers, and to provide the resources that are required to make progress. So some of the treatments that we give today are based on research that was done 10 or 20 years ago. Some may be based on research done only a few years ago, but we need to make investments in future therapies now, even though we are also innovating in terms of the drugs that we have available, the way we combine drugs, the strategy of treating or not treating. Even before I was at Sloan Kettering, in my early days, I used to treat multiple myeloma on the West Coast. And at that time, the prognosis was usually about 3 years was the average survival for a patient, 3 to 4 years. Lots of drugs were being combined. No progress was being made. And Ola, Dr. Landgren will fill you in on some of the amazing things that are happening in the area of myeloma. What I'd like to say a little bit is about our journey as a cancer center. So in 2019, we received the NCI designation, the National Cancer Institute designation. We were the 71st cancer center to receive the designation, the only one in South Florida. Yesterday, the University of Rochester's Cancer Center just became the 73rd in the United States, and I can tell you that our grant still has received the best score of any new cancer center grant in the last 25 years. So it's a reflection of what the National Cancer Institute thinks is the quality of the work that we do at our institution. Dr. Landgren has been here about 4 years, and I remember when we had a dinner to talk about the possibility of bringing him here, and I think that we've forged a partnership over the years. We share a few qualities. I think we're both relentless and proud of that. We both have very high standards and are proud of that and insist that everybody live up to our standards. I don't think we're very compromising, but I think we also, and I'll just talk about him because I can't really say about myself, but there's a level of humanity and of leadership. And so before Ola came, we had one really outstanding multiple myeloma doctor, and now we have something like 11, and hopefully more to come. And people want to come to a place where they see a future for themselves. And so under his leadership, we formed the Myeloma Institute. We've recruited all the people that are on this stage and a number that are in the audience, and we're just getting started. But I think it's an important, what Ola and the team have done for the Myeloma Institute is very important for South Florida. So we have maybe the second best, maybe we're in the top 3 in the world in multiple myeloma, and that's a very recent thing. And wherever we are, it doesn't matter exactly because next year we're going to be better. And what's happened as an NCI-designated cancer center is that there are therapies that are only available at our cancer center, and so that's very important. And we have a very important mission to provide the best care, the most compassionate care, to be available. Dr. Kazanjian at the other end, I called him up, I think it was on a Saturday, to talk about a patient, and then he agreed to see the patient on Monday. Okay, and so that he was available on Saturday is typical. Okay, it's not something so special, but you may not know that it's not special. But not every doctor. My own doctors don't seem to be available if it's 1 minute after 5 p.m., but I do have most of their cell phones. But, it's very important, the whole package, and everything matters to us. And that's why we continue to attract people, patients, the numbers every year. We're seeing more patients, which is good because these guys really know what they're doing, okay? And not everyone does. I would say that in the hematologic cancers or the blood cancers, somewhere around 15% of patients, maybe a little bit more, are misdiagnosed. And so they're either told they don't need treatment and they do, they're told they need treatment and they don't, or they don't even have the right diagnosis. And so, getting a second opinion and seeing real experts is just fundamental. We have benefited greatly from our community that we work in. We just recently had our bike ride and the Dolphins Cancer Challenge, and this is our 15th year. When it started, there were a couple hundred people who came out. And this year, there were 7,300 people who participated. And we met our goal. We've raised over $15 million from the bike ride, which is pretty remarkable. And more importantly, there were over 120 cancer survivors who participated in a program called Believe in You. So when we started, the patients didn't participate, but the family members did. And now the patients are participating. 2 years ago, I rode 100 miles, and along the way was a guy who was in the middle of his radiation therapy. So on Friday, he got radiation. On Saturday, he rode 100 miles, and then on Monday, he started another week of radiation therapy. A tremendous inspiration to all of us, and certainly an inspiration to any patient who is wondering about, getting back into their normal life again. And so the other comment I would make is that we're opening up a new cancer research building in about a month. And we received last year a $50 million naming gift from Ken Griffin, who is a very wealthy hedge fund guy, one of the best investors in the world. And I think that he wouldn't have invested $50 million in us unless he thought it was a very good investment. And so we're very grateful to him and the Citadel people. The building is a symbol, the $50 million is a symbol, but what you're going to hear tonight is tremendous expertise in caring. Both are equally important. And we're thrilled to have you as a captive audience tonight. And so I'm going to turn things over to Dr. Landgren now and have him talk about himself a bit and the Institute and what he's accomplished and what he wants to accomplish, and then we'll go down the line here. Ola.
Thank you very much, Stephen. Thank you very much, everybody, for coming. It's really an honor having you here. So I see very many familiar faces here. For those of you who don't know me, I'm Ola Landgren. I'm MD, PhD by training. I've been here for about 4 years, and I'm the leader for the Myeloma Institute here at the Sylvester Comprehensive Cancer Center. I'm originally from Sweden. I came to the United States 21 years ago. I thought I was going to spend 2 years at the National Cancer Institute in Washington, D.C., and then return back and continue building a program back in Stockholm, Sweden. But I was offered to apply for tenure track, and then I was tenured, and I stayed at the NCI for 10 years. There was no myeloma program when I came, so I was offered to build a myeloma program, and that was a lot of work, so I thought I would probably stay here for the rest of my life. I had a lot of job offers, and eventually, many years ago, Sloan Kettering came and knocked on the door and said, "You want to come to New York City?" So I was thinking, maybe I should do that. So I went there, and I was offered a job, and it turned out they did not have a myeloma program either. So they said, "We want you to build a myeloma program. We have pretty much every other program, but not myeloma." So I took on the job, and this was in 2014, and I built up that program, and it was a lot of work. And in 2019 or so, we had over 10,000 outpatient visits. We had operations in Manhattan. We had research labs. We had clinical trials. We have a very strong program. I thought, I'm going to stay here for the rest of my life. But I've known Dr. Neimer for a long time, and he's tried to recruit me many times to come to Florida. I said, look, find someone else. I'm not leaving New York City. But in 2019, 2020, I came down here, I think, for the second or third time. And then on the way back, when I came back to New York, I said to my wife, I think we are ready to move. She said, where? To Florida. She said, let's do it. So, so Dr. Neimer recruited me to come here. So I started here officially November 1. But I said to Dr. Neimer before I signed the contract, be careful when you hire me because I'm going to ask for a lot. Because I want to build one of the top 3 programs, because this will be my 3rd program I build. And I heard someone said, when you build a house, the first you build for your enemy, the second you build for your friend, and the third you build for yourself. So I've done so many mistakes, so I'm not going to do those mistakes. I'm going to do other mistakes. So in record time, I was employee number 4. That was Dr. Hoffman, who's a fantastic doctor. He's also trained at Sloan Kettering. He was here. He's nurse practitioner, Susan. She's from Brooklyn in New York. And they had a nurse or a patient coordinator, Gigi. And then I came as number 4. And then it was 5, 6, 7, 8, 9, 10. And I think we're over 60 people now, something like that. And that takes a lot of counting. Actually, I interviewed someone on the way here, so I'm going to hire more people. So I think our right size for our organization is probably somewhere a little bit over 100. Because there are so many patients that need help here in Florida. We don't want anyone to have myeloma, but Florida has the highest number of myeloma patients in the United States, and there are around 20,000 people living with the disease in Florida. We also have a lot of patients traveling here. Actually, on Tuesday, I had second opinion from New York coming to Florida to see me for second opinion. People fly from Manhattan to Miami for second opinions. It used to be the opposite before. So we have a lot of patients coming here. So we are building on high quality. We have doctors, nurses, nurse practitioners, pharmacists, social workers, patient coordinators working in small teams. And I learned this at Sloan Kettering. If you build a team like that, you can really go to 100% quality. And when that team is starting to get oversubscribed, you hire another team with a new doctor, nurse practitioner, pharmacist, patient coordinator, social worker. And you build small groups like that so they can cater top-of-the-line care for patients. That's how Sloan Kettering is built, and that's how we are built also. And I also learned at Sloan Kettering that it doesn't matter how good your care is, how smart people are. If you're not where patients live, that becomes a problem. Patient came to me in Manhattan and said, tell me about what's going to happen next. And I went over everything and said, do you have any questions? I have two questions. Where do I park my car, and where do I eat lunch? And I think here probably more people are asking, how will I be able to drive here? Because going from Aventura to here, or from Deerfield to here, is just not possible. You can do it one time or two times, but you couldn't do it once a week. So we are also setting up service in all the satellites. So I'm now in Coral Gables. I started in Miami downtown, but I'm now in Coral Gables. I'm there with Dr. Kassanjian. We have Dr. Diamond and Dr. Pandy and Dr. Hoffman downtown. We're going to open up very soon in Doral after the summer. We just hired a doctor from Boston here. And we are also opening up in Solomia in Aventura. It's going to happen later this year. Hire another doctor. And we are also in Deerfield. So having the best possible care built with optimal processes that can deliver 100% for dedicated team to individual patients and have that operation where the patient lives. That's the model. We're also taking all the latest drugs here. So we have over 20 trials. So Michelle, who sits here on the first row on my left side, she also came from Sloan Kettering. We cannot call Sloan Kettering anymore because they're worried we will steal people. So we're taking so many people from there. So Michelle is our director for research. So she oversees all the clinical trials. We have 20 open trials. And we don't have those trials that randomize between a new therapy and an old therapy. Because why would we expose our patients to that? We will only open those trials that have the newest drugs without randomization. When there is something that's unique, if there is no other option for that patient, or this is a better option for individual patients. Those are the trials we have. So we have all the FDA-approved drugs, plus that we have the additional drugs that you cannot get elsewhere because they're not yet FDA-approved. And we're going to open more, so we're going to probably have around 30 trials open by the end of the year. And that is really giving access to the latest. We also have MRD testing. We have profiling in the blood and the bone marrow. We are developing blood-based tests so we don't have to do biopsies of our patients. And we're also developing new imaging technologies. We're trying to understand mechanisms of response and resistance, biomarkers, and we're basically covering all the bases. We have a very, very strong team, and I'm really happy here, and I'm blessed to be under the leadership of Dr. Neimer and all the support he's giving me and our team. I would like to hand over to Dr. Diamond.
Continuing on the theme, I met Ola at Sloan Kettering. That's where I was doing my fellowship training. And so I've been working with Ola for the past, 6 years at this point. And I was a fellow down there. I was really only in clinical medicine at the time. And he showed me this new world of translational medicine, where you could actually use a bunch of new technologies to fully understand disease, to try to personalize medicine a little bit better, and to deliver better care. And so since then, under his leadership and his support, I've been working a lot on genomics. Okay, so this is trying to understand what makes cancer tick. In this case, the myeloma. We're looking at the DNA of the myeloma and lots of techniques to try to fully understand the breadth of all of the mutations that we're seeing, what makes everyone's disease unique, and how we can tailor our therapy best to attack all of the lesions that we find in these unique diseases. So at this time, I have a day of clinic a week where I'm seeing about 30 to 35 patients on a weekly basis. We have a great team downtown. I work with a number of these lovely people that are sitting in the audience, and I couldn't do it without them. And the bulk of my time actually is spent both running clinical trials and then working on research to fully understand the disease. So again, I said I work on tumor genomics. This is trying to understand what makes the disease tick, looking at DNA. All of our patients are generally being subject to a lot of these tests. We have a number of experts in the room that are looking at different aspects of what makes myeloma go. So you'll hear from a couple of them today. We have people in the audience that are working on trying to understand the tumor microenvironment. So aside from looking at the DNA of the tumor, trying to understand the immune system and what allows the myeloma to grow. And you can actually hear from them later if you talk to them out in the audience. But generally speaking, this is what we do. We're super happy to be here. Ola's built a great team, and I'm happy to show up to work every day working under his leadership.
My name is Katie Thorne. I'm a little bit different here. I'm a PhD clinical chemist in the Department of Pathology. So I actually don't see patients, but I see your samples. So I oversee 2 labs here. So I oversee the clinical lab where we do the routine myeloma testing, so serum protein electrophoresis and immunofixation, free light chains. I also oversee a translational lab for the Myeloma Institute where we're developing and evaluating new diagnostic tests. So I guess, to follow the theme, a little bit of my background, I got my PhD in chemistry from Berkeley, and I did my clinical chemistry fellowship training at UCSF, and my first job out of training was at Memorial Sloan Kettering. So that's where I met Dr. Langren, and I actually specialized in mass spec training during my fellowship. And around that time, they were developing new mass spec assays for myeloma. So I remember meeting with Dr. Langren and saying, hey, I could potentially set up some mass spec assays for detecting the low levels of the M protein. And he just said to me, do it, yes, do it. So so we worked together for, I think, 6 years at MSK. And then I've been here at University of Miami for 3 years, so.
I want to echo everyone and then thank you guys for coming. It kind of means a lot for those. I know some of you and some maybe in the future we'll meet. But to give a little bit of my background, I took sort of not the typical way, as is like some other folks. But I grew up in New York. I never went back actually after high school. I got my training in Boston, and I was in the Air Force for about 6 years. And ultimately went to a fellowship to learn hematology oncology at the National Institutes of Health, that's where I met Dr. Langren, and that's where our connection comes. Everyone assumes it's the MSK, but I actually never worked at MSK, Memorial Sloan Kettering. And I stayed there up until about a little after Dr. Langren arrived. And at that point, I was leading the program that Dr. Langren had devised or created at the NIH, and I also had a dual appointment at the FDA. So a lot of my career path had been, drug development, early phase trials, regulatory work, and really try to push the envelope to get new drugs, out sooner to patients. And so, Dr. Langren gave me a call and asked me what I was doing, and then I guess a few months later I was in Miami. In fact, I had accepted the position without ever being in Miami, ever. I'm not talking about a job interview in Miami. I'm like, I'd never seen in Miami. But that's how much confidence I had in Dr. Langren, Dr. Neimer, and the program. And then so, my role really is to support Dr. Langren and the myeloma program in devising what we call investigator-sponsored trials. I know my patients know it, and I'm sure all of you know it, but just to remind you, the investigator-sponsored trials are sort of the unique opportunity for clinician scientists who have hypotheses, who have a thought, who want to test their thought out. That's when they write a clinical trial, and and it's very difficult because you have to find the funding source, you have to convince the drug companies to give you the drug or whoever that's going to help you. But it's actually your baby, and that was my, I came with the idea that that's what I was going to work on and broaden that enterprise. Also, given some of my other sort of unique experiences, I also try to help not only the myeloma program, but the other investigators in the other sections in trying to get things through, the Food and Drug Administration or the IRB to write clinical trials, protocols, et cetera. Thank you.
Okay, so I think everyone's been able to introduce themselves, and hopefully you understood most of that. And what I'd like to do is just start by asking Dr. Langren the first question, and he's going to know the answer. Okay, so let me start out. He is going to have an answer. So, you've been in the field for a long time, and you've seen the therapies evolve. but I know one of your sort of, I can't say pet peeve exactly, but something that you've talked about a lot is how even though the disease is different in everybody, that oftentimes the approach initially is the same for different people. And so can you talk a little bit about how the Sylvester Myeloma Institute thinks of precision medicine and how you think about incorporating all the data that you collect into deciding who gets treated and with what?
That's a very good question. So yeah, I have thought about that a lot, and I thought about it at every place I worked. But I think we are now at the juncture where we actually can deliver because the technologies are available. We can profile individual patient samples with sequencing. Sequencing used to cost millions of dollars and take 10 or 20 years. Years ago, that was the Human Genome Project. Now you can do sequencing in a few days, and it could technically cost maybe $10 or $100. It's not yet there. We still have to pay a little bit more than that, but the prices are going down. Every year, they just keep on dropping. So what we are doing, we have opened a study called the Flagship Study. So we are doing this as a pilot study, and we hope by the end of this year that study will be completed, and that will then have all the data generated for patients on that trial, and we will compare it to all the current standard of care. And we already know, we have preliminary data showing that it will replace all the old technology, and all the new technology will give us all that extra information. What that means is basically you switch from a black and white old TV to a high definition color TV. You can see the individual patient's full color for every aspect of the disease. We think that myeloma is probably at least 15 different diseases. And we think that the next move will be to start differentiating the different drugs that we use. This will have to be done in a stepwise manner. We know that all the drugs we have today are very good, but there could be individual patients, but there still could be some residual disease cells left. And that's how we're going to use this technology to get rid of every cell in those patients. My ultimate goal is to develop a cure for myeloma, and I think we will do it. And lastly, I think profiling is important. The drugs we have now are very, very good, and there are more drugs coming. We work very closely. I mentioned we have 20, soon 30 trials open, and those trials we can get here for these investigator-initiated studies that are really built to test the hypothesis and try to build a cure. I think the last piece in the puzzle that we have worked on for a long time, I worked a lot on minimal residual disease detection, MRD. So if you can test a patient and with very high confidence say there is no disease left, and if you can do that a few times, that is the way towards a cure. So I think profiling, optimizing the use of the drugs, and do measuring and ruling out residual disease, that's how we are pushing the envelope here at Sylvester. So we have all these technologies, and we're going to just keep on making it standard of care here.
Yep. So more drugs available, better outcomes, which always raises questions about the quality of life. We were just talking about the importance as you get treated that you can be relatively free of side effects or completely free of side effects. And I know some of your work has been trying to understand some of the side effects that the different drugs cause. I know some of your work is about learning how the drugs, when they stop working, what's happened. And can you talk a little bit about your approach to, one, getting us towards drugs that are effective with less side effects, and then also how do we make these drugs work longer? So sort of a two-part question.
Sure, I'll do my best. So thanks for that really interesting question. And it's very true. We have great therapies right now, but not all therapies are right for every individual. And sometimes we are offering treatment that is sort of over-treatment or overkill, and we're suffering with side effects. And we'd really like to figure out who needs an escalation of therapy and who can get a little bit less. So one of the things that we've studied in the past is a side effect that is really horrible. This is a therapy-related leukemia. This is where you give something like a high-dose melphalan transplant, which is a standard of care in myeloma where people are getting high-dose chemotherapy and a stem cell transplant. And it's very commonplace. And one of the most feared side effects is that a second leukemia develops a few years later, just because of that intense mutagenic activity of the chemotherapy. So some of the research we've done in the past is trying to figure out exactly who are the people that might be most at risk for this. And we have found that actually people that bear pre-leukemic clones, in other words, sort of small precursors that are already existing in the blood, might be at risk for this condition, and specifically clones that have specific mutations. And we've been able to show with sophisticated genomic machine learning algorithms, basically, that it is the chemotherapy itself that's inducing those clones to further on develop leukemia. So at this point, we're basically trying to make sure that we're only using the therapy that is needed for the individual patient. And so when every patient comes in, we're trying to understand their disease, understand their risk, use MRD tests, as Ola said, to try to tailor our strategies and make sure we're not doing too much in patients that have already achieved very good outcomes. To your point on the second, trying to understand resistance. We have fantastic therapies, more coming every day. The armamentarium that we have is really to be jealous of among all of the other diseases. But unfortunately, people still do experience a relapse. And so what we've been doing is using genomic techniques to understand exactly why that happens. Sometimes we get mutations that make basically the myeloma invisible to a lot of our immune therapies, and it allows them to escape. And actually what we found is that some of these mutations actually can be overcome by just switching drugs to sometimes even agents in the same class. And so we've been working on that a lot. More importantly is how do we identify them faster and earlier so we can make the change quicker. And as Ola said, we are working on some techniques to measure these things from the blood so that we're able to enact these changes faster. We're actually working on some data as we speak that will help us track some of these mutations in the blood.
Thank you. Dr. Thorne. Dr. Landgren mentioned minimal residual disease, which basically means using some techniques to find the very tiniest amount of disease to determine whether someone is really free of their myeloma or not. And you talked about mass spec. Maybe I can ask you to talk about the basis for the different tests for MRD, and maybe you could explain what mass spec is, because maybe everyone doesn't know that yet.
Sure. Okay. So minimal residual disease, I think the current accepted tests right now use bone marrow as the sample type, and you're looking for one myeloma cell out of 1 million. So it's a really sensitive way. Usually it's done by either flow cytometry or next generation sequencing to detect that rare myeloma cell out of the normal cells. The downside to it, obviously, is it needs a bone marrow biopsy. So what we're really focusing on here, and it's already been referenced a couple times, is developing MRD tests that use blood samples, so blood-based MRD. So there's a couple different approaches that we're taking here at University of Miami. Mass spec is one of those approaches, so it's called mass spectrometry. It's basically an instrument that measures the mass of compounds. And what we do, we're looking at the monoclonal protein, so the protein that's produced by the myeloma cells. And we can measure the mass of that protein, and that's specific to the patient's myeloma. And so once we know that specific mass, we can track that over time, and we can track the concentrations go down to really low levels, sometimes go undetectable, and then we can also track if it comes back up in concentration. So it's just a technique that's used that's a really sensitive way to measure low concentrations of the monoclonal protein. Certainly there's other techniques that are used for blood-based MRD. There's looking for the DNA in blood or looking for circulating myeloma cells in blood. And we're also looking at those techniques as well. And so I think, yeah.
Dr. Kazanjian, in the past, most of the routes of therapy ended in an autologous stem cell or bone marrow transplant. And I know that that's something that the whole field would love to be able to move away from. Can you talk a little bit about some of the recent strategies that are being used to treat myeloma that may, in fact, over time replace the autologous transplant?
Thank you. So I think a couple of decades ago, autologous transplant was very important, but it wasn't the transplant part. It was really the high-dose melphalan part, because it was sort of the only therapy that really worked in myeloma and started to change the natural history of the disease. But that was a couple of decades ago. And so since then, we've had a lot of drug development, a lot of better drugs. And so I think that paradigm where everything has to sort of be around autologous transplant has changed. It's not is changing, but has changed. And what we do know are a few things. So when we, based on some of the studies that actually this group has done, using 3 and 4 drug novel combinations, patients are getting into that MRD negative remissions like Dr. Thorin was just speaking about. And in those cases, and Dr. Langren has done a lot of this work, is you probably don't need transplant if you can get that deep of a remission. So then the question really becomes, well, what if you don't get in that deep in that remission? What do? So do you just sort of live with it, or do you actually go and get the high-dose chemotherapy with the autologous support? Or is there a third option? And that's what we've actually really tried to work on. And one of the things that we've been working on is using the newer immunotherapies to do that. And so we do have an investigator-sponsored trial that's open and it's very preliminary, but the results look very good. And obviously the side effect profile is nowhere close to high-dose chemotherapy.
Okay, thank you. Back to Dr. Landgren. And I'm going to go to the questions from the audience. So someone asked, what is the best treatment for multiple myeloma? And I know that you've recently published sort of a guide that can be used by physicians around the country so that they can, based on all the data that you've accumulated and analyzed with your team. But can you talk a little bit about how doctors determine what's the best treatment for myeloma and your recommendation for how they should be doing that?
That's another very good question. So maybe I answer the question, how is myeloma treated today, and put it in the context of how it was treated a few years ago. So today, many patients can be treated without chemotherapy. And when I was in fellowship many years ago, chemotherapy was the only thing we had. We had combinations of chemotherapy followed by high dose of chemotherapy, which is what Dr. Kasanian was talking about. In the United States, it's called transplant. In Europe, it's called high-dose chemotherapy. It's the same treatment. You give chemotherapy, and then you have harvested the patient's own stem cells before chemotherapy, so you give them back afterwards. But the cells have nothing to do with the treatment. It's just to tolerate the high dose of chemotherapy. Today, we use small molecules, we use immunomodulatory drugs, and we use antibodies. So someone who's newly diagnosed is treated outpatient without chemotherapy. And we can deliver probably remission in 98 or so, 99% of the patients taking that approach. Used to be not the case. When we gave chemotherapy, when I was in fellowship, maybe 20% of patients reached remission, if that. Now it's 99%, no chemotherapy. There are new immunotherapies coming. We have new antibodies. There are new other type of approaches. Everyone knows about the COVID vaccines. They are mRNA-based. There are new mRNA-based technologies in development, and we are one of the selected sites. So when this is ready to go into humans, we will start using it for our Myeloma Institute. You can get an equivalent of an injection, and the injection can make the drug in the body. And there are other technologies as well. So it's all about the immunotherapies and the immunomodulatory drugs and the small molecules, I think. When it comes to dictates what individual doctors choose in terms of combinations, The FDA obviously has a big say, because if the drugs are approved or not, that will dictate what you can do. But there's also something called the National Comprehensive Cancer Network, the NCCN network. I used to be a member of that when I was at Sloan Kettering. I used to be on the committee that dictated which drugs were approved in the United States. I have written 2 protocols that have ended up in that committee guideline, and these are trials. We did investigator-initiated studies. So we developed a trial, and the results were very successful, and the committee deemed it so successful, so it became approved and therefore accessible for patients across the country. So we have contributed to that. So that guideline is also dictating in parallel with the FDA. Lastly, Dr. Nairo is asking about the work we have done with integrating biological data, like genomic data, with treatment data. So in a simple way, we have taken information on about 2,000 patients, where we have information on the genomes of the disease, and we also know how the individual patient, how the treatment was given. And we also know how the prognosis was for that individual. So instead of looking at the average, say that among these 2,000 patients at 8 years, this was the percentage of freedom from the disease coming back. Instead of doing that, we used, similar to ChatGPT, we used machine learning, artificial intelligence, and that allowed us to ask the computer in this database with all these individuals, over 2,000 patients, what would be the optimal way to treat a patient that looks like this with the biological workup, if you compare to the database. And I think that's how the field is going. So we are trying to work very much along these lines to take advantage of new technologies. So artificial intelligence is something that we are integrating more and more in our research.
Thank you. You actually anticipated my next question, which was, someone asked, how is AI incorporated in the Myeloma Institute? Ben, you may want to comment a little bit about that, but also can you talk a little bit about collaborations with other institutions? I'd say one of the questions that I'm always asked is, do we talk with other doctors and share information, or is everything kept a secret?
Both great questions. So Ola already told you a little bit about our AI-based approaches. So we're using them in a lot of different ways. On an individual basis, a lot of us that are doing the genomic research, that are looking at a lot of data, huge data sets, are using AI to help us code better and to be more efficient and to write better code. So all of us are doing that. So it's really sped up our research. To speak specifically about a project, for example, that we've used it for, as Ola mentioned, there was a very large effort to amass 2,000 patients and their genomic information from many institutions. Okay, so this is a huge collaborative effort, working with Heidelberg University in Germany, working with Sloan Kettering, with the Mayo Clinic, et cetera. We have a lot of friends out there. And we compiled all this data and used it to try to understand the biology behind myeloma, and actually reclassified the disease into 12 molecular subgroups. Now, the nice thing about the machine learning approach that we were able to use afterwards was that for each of those patients within a subgroup that, first of all, had never been described, we were able to ask the computer, as Ola said, what therapies are going to work best. We could simulate what the outcomes were going to be and make sure that we understood which patients were getting the best therapy. Now, this can be used to obviously choose and tailor therapy that might, be strong, but it can also be used to figure out which patients don't need certain therapies, what might be overkill. And so that model's been published. It was a relatively large effort, and actually the tool is available now. It's online, and anyone that wants to use it can do so. It's difficult to use now because not everybody's using genomic testing just yet, but it's becoming much more commonplace, and this is a model that can be grown in real time and added to. And so it will continue to grow. It'll have the new drugs added to it, and we'll be able to use it much more often. And so again, as Dr. Neimer said, we do collaborate a lot. That took a lot of people to work on it together, and to this day, we work very closely with a lot of different institutions around the world because we're all interested in doing this together.
Thank you. Katie, you talked about blood-based tests versus the wonderful bone marrow biopsy, which some of our patients have to undergo. How close are we to eliminating bone marrow biopsies? And
Yeah, that's a good question. I think so. We've made a lot of progress with the mass spectrometry tests that measure the really low concentrations of the monoclonal protein. We are still trying to understand the clinical utility of those tests, basically when it's appropriate to use those tests. And we do a lot of comparisons versus the bone marrow-based MRD tests. So they do correlate pretty well, but I think we do see some differences, and we're still trying to figure out what it means when you have a protein at a very low concentration, yet you might be negative by the bone marrow. So there's occasions where the bone marrow could be, you've just missed the disease in the bone marrow, or there's cases where the protein sticks around longer, yet you don't have disease still. So those are some of the things that we're still working on. We do that incorporate those tests in big clinical trials so we can see which one is sort of a better predictor of patient outcome and how the patient does. But I think probably still a couple years away, and there is still useful things that you get from bone marrow biopsies that you can't get from measuring just the protein. But I think the mass spec and some of these blood-based tests can help reduce the amount of bone marrow biopsies, right? So like one of the advantages, the fact simply that blood draws are much less invasive, much less painful, allow us to sample more frequently, right? It's much easier to do a blood draw more often, right? So we can track patients more closely and sort of watch the trajectory of that protein, whether it's going down or whether it's staying stable, and that could inform us of when a bone marrow biopsy is needed, or maybe you could hold off on that. So hopefully, I think it would reduce the amount of biopsies that are going to be done.
Thank you, Dr. Kazanjian. So, Dr. Landgren talked about the different types of clinical trials, sort of the ones where you sort of flip a coin and decide, the coin flip decides on the therapy, which, we don't really do that, thinking that there's better ways for patients. Can you talk a little bit about the sort of clinical trial infrastructure that's required to support these 20 trials, and a little bit about the importance of being on a clinical trial if you were a patient?
I can speak all day about that, Dr. Neimer. I think I'm very fortunate to have a very strong research team, as I think we all are over here. And the doctor just walks into the room for, let's say, a clinical trial patient, but there's probably 20 to 30 people working behind that doctor to make sure that between the prescriptions, the labs, the coordination with the actual different manufacturing to get the drug, the FDA stuff, it's enormous. And that's why Dr. Landgren said is we need 70 people for 4 doctors, to do the research. And I think research is important for us, obviously, to advance the field. Research is important for patients because not only because they really, you guys who are in the research are really doing a public service to the community, but also it's been proven you're actually getting better care. And even with us, like time and time again, research has shown that because of all the oversight, because there's one doctor that walks in the room if you're on the clinical trial, but there's 4 doctors in the back room that are reviewing your chart that you don't even realize. And so I think it really, like, everyone is benefited from being on a clinical research study. Thank you.
Okay, so I'd like to take just a moment, and I know that everybody's not here and people were not expecting, but everyone in the audience who's part of the Sylvester Myeloma Institute, if you could stand for a second and everyone can see. So it takes a huge team. I would also say one of the things that makes the clinical trials so important? One of the questions, and Ola addressed this, we don't really talk about a cure yet for multiple myeloma, but we're relentless in pursuing a cure. And our hope is that the next clinical trial, which is building on what we know now, will be able to eliminate the disease, and that when we're done with the treatment, the disease won't come back, which is really what a cure is. And so I think it's the clinical trials that we are opening or have opened are designed to help people live better and longer and ultimately cure the disease. So I have a final question for Dr. Landgren. I'm going to combine two of the questions from the audience. One person said, I don't want to get too political, but how is the uncertainty around federal funding affecting the research? And the second was a question, what are the funding priorities for Sylvester's Myeloma Institute? So if you could sort of tell us what are your priorities and what we should do to help you.
Thank you. Those are difficult questions. So, well, when it comes to the current change for funding, everyone can see online and read everywhere about all the changes. So, I mean, there's always change. So, having been around for a long time in the field, I do expect change, and we do expect change all the time. So we build our program so the program can take change. It's like if you have a boat that's going to go out on big oceans, you better have a big boat. So that's sort of how we build our ship here, or boat. But of course, everything matters. So when the federal funds go down, we have to rely more on other sources. So we work with foundations because the research will require financial support. Otherwise, we cannot do it. So we rely on foundations, and we also rely on philanthropic donations and other types of sources, different types of contracts that we can have. So it's sort of a mix of these things. And we build the program, as I said, intentionally so we can handle changes in any of these sources. But one thing that will never change is our mission. Our mission is to continue, as I said before, is to seek and develop a cure for the disease. That has not changed, it will never change. And we will keep on working relentlessly, whatever all those other things are. When it comes to our priorities, I would say the model I try to build, my leadership style, building the Myeloma Institute, is built on people. I look for talent. The people, I try to put together processes, because processes are efficient. That's fuel efficiency. So people and processes are my first two P's. And the third is priority. And the priorities will always change. And as we grow, I was, as I said, employee number 4. And now we have over 60 people. So of course the priorities have changed over time. So when it comes to funding priorities, I would say that the overall goal for the program and the direction is aligned with the Cancer Center. We want to develop precision medicine and MRD testing. We want to develop sophisticated translational research studies. We want to build team science, and we want to be active in the clinical trial arena to deliver the cure. But those are sort of the big picture priorities. The smaller priorities could be that we want to make sure that individual investigators have sufficient support to do their daily job. On next level up, I want to make sure that there is a critical mass of people that do certain tasks. And then on a higher level is really to drive the program and make sure that we have program representation in different locations. So to me, it's sort of a multi-level process, but everything comes back in my head, people, processes, and priorities. That's what I follow. That's my map.
So I'm wondering if there's either a caregiver who would like to say anything before we wrap up, give you an opportunity, or yes. So let's...
My question is regarding the treatment and the CAR T cell therapies. So what I learned and I read is that at the beginning of the therapy, you have more T cells to be able to mix with the new medicine that you put back. As long as you go with the treatment, those T cells die, and so you have less. So my question to you, doctor, is why is the CAR T cell, I don't know with you, but in general, used as the last kind of, I know it's very expensive. That's my question.
So thank you so much for asking the question. So the question is, why are CAR T cell therapies used for later line? Well, in fact, that has changed. In 2020, the first CAR T cell therapy was approved for the treatment of multiple myeloma. And in 2021, the second treatment was approved. In 2024, in the spring of 2024, the FDA approved CAR T cell as the second line. So someone can get combination therapy and can then move on to CAR T cells. So the FDA has actually changed that. It's less than a year. There are also ongoing trials, including a trial we have here at Sylvester Comprehensive Cancer Center, where we have CAR T cells for newly diagnosed patients. Now, is CAR T cell the only solution? Well, it depends on who you ask. If you ask the people that really believe in it, they would say yes. But there are people that believe in many other things. And if you just look at the data, then bispecific antibodies that are T cell engaging, what they do is that they move the T cells inside the body. Instead of taking them out and making CAR T cells, you can move them so they move closer to where the myeloma cells are, and then they can kill the myeloma cells. Those we also have. We have many clinical trials. We have trials here at Sylvester for smoldering myeloma, for relapse myeloma, even for the first relapse. And we are just about to open such a trial for newly diagnosed patients as well. So there are a lot of options, and it's no longer for later line.
How y'all doing? Okay, I want to know about a patient that has aggressive multiple myeloma that every week, every time they do capillary change, the numbers increasing, hundreds, 200s, and they try different medicine, but the medicine is not working. And so they got this treatment where they give patients, in case of that emergency, where they send them to the hospital for a couple of weeks and they give them EPOT. EPOT kill all the cancer cells, but it also kills a lot of the good cells. So I want to know, are y'all working on the treatment that eliminate, even though if the cancer more than likely might come back, but is y'all working on the treatment that if somebody has aggressive, if somebody multiple myeloma is so aggressive that none of the treatments are working, instead of EPOT, because EPOT have a lot of bad side effects, are y'all working on something that eliminate the cancer, even though it might come back quickly, but if somebody, let's just say somebody capillary change is 200 and then they go back to the doctor, they capillary change in the thousands, then they're going to 5,000, 10,000, 14,000. The EPOT was made to eliminate the cancer, but it also killed a lot of cancer cells, which people be prone to infections and other things happen in the process of that. So I want to know, do y'all have another treatment that y'all working on besides the EPOT with the bad, with the bad side effects?
I think that's a great question. We really try to reserve EPOC, and EPOC like DCEP is another regimen, and there's something called VTD PACE. And for those who don't know, these are just acronyms for kind of the more traditional cytotoxic chemotherapy, which we really, as it kills all the myeloma cells, also lowers all your normal cells. And when that happens, it's just like the high-dose therapy with melphalan. Patients can be susceptible to diseases and other things. In myeloma, we generally try not to do that up until sort of at the end of the management course, if need be, that is. Really, patients, for the most part, would have had to exhaust most therapies. There's select cases where if you have very aggressive and you just need to get a hold, of the treatment, then really we don't have anything to replace that per se, but maybe someday we will. The problem is a lot of the great therapies that are coming out, the immunotherapies, whether they're bispecific or CAR T, they always work better when you don't have a lot of multiple myeloma. And so that's why one of our sort of ways we treat this is to try to what we call debulk or get that initial tumor down with sort of this things like EPOC, and then use the immunotherapies kind of do the final sweeping on the ground. But I mean, I think that's an active interest area, and hopefully, you know, in not so long we'll have therapies that won't harm our normal cells as much.
Okay, so what I'd like to, in closing, just to say a couple things. So multiple myeloma is one of the types of cancer where there's been incredible progress made. There are many more options, whether it be these bispecific antibodies or CAR T cells or other immunotherapy approaches. Some of the very most modern approaches, something called a proteasome inhibitor, was first introduced in multiple myeloma. And so the treatments continue to evolve. What's also important, and there are people who are diagnosed who don't need to be treated. They're at a stage where the treatment hasn't been shown to work and they're observed for a while. And I think Dr. Landgren has done with his colleagues some pioneering work in figuring out what's called either MGUS or smoldering myeloma, and to try to understand why people over time develop myeloma and others don't. There are other diseases. Amyloidosis is a disease that's related to multiple myeloma, and there's great expertise there as well. There are also diseases called plasmacytomas, which are related to multiple myeloma, and that the expertise in the group extends about all of these diseases that are part of a family. Everyone's disease is different, but by accumulating data from thousands of patients as they've done, they really are able to pin down who needs what therapy, and also to understand as early as possible when something isn't working. Multiple myeloma is one of the cancers that can affect a lot of other organs in the body, the kidneys in particular, and infections and like pneumonia. And so catching the disease early as it's coming back is also a way to prolong life. And so I want to thank the group here. We have...
Can I ask you a quick question? The fish marker 14, the marker,
Yes.
do they still use it? And how accurate is it?
Okay, do you want...
So the question is the fish marker for chromosome 14, if that's still used?
Fish 14.
Yes, it is still used. It was developed about 40 years ago. I think it's eventually probably going to go away because it's going to be replaced by the type of technology I mentioned before. We do the flagship study here at the University of Miami at the Myeloma Institute, where we are using sequencing. Chromosome 14 is an important chromosome, so there are 23 chromosome pairs in the body. There is one chromosome from your mother, one from your father, and we all are born with 23 chromosome pairs. On that 14th chromosome, there is something called the IGH locus, which is a region in the genome that regulates activities that are relevant to what's going on in a plasma cell. And myeloma is a plasma cell. So in about half the patients that have myeloma, we see that on chromosome 14 there are some errors in the code. We think that is how myeloma started in those patients. And when we look a little bit closer, we see that chromosome 14 in some patients have swapped information with the fourth chromosome. We call that translocation 414. In some cases, it can be swapped with chromosome 16. We call that chromosome 14-16. Sometimes it can be chromosome 14 and chromosome 11, 11-14 translocations. And it can also be chromosome 20, 11-20 chromosome. So why these particular chromosomes? Well, there are actually something called oncogenes. There are some genes that are bad genes that live on these chromosomes, the other chromosomes I mentioned. And because chromosome 14 contains the IGH locus that's very active in plasma cells, it seems that in these cases, when this happens, that the driver for plasma cell has partnered with an oncogene. And that is probably how those myelomas started in the first place. So that's why there has been interest in studying chromosome 14 for a long time. But if you use FISH probes, there's a panel that's used in all the labs around the world. It was rolled out about 40 years ago. You have about 20 markers, and you can test and see, does this patient sample have this marker, yes or no? That's pretty much how FISH probes work. You can get up to 20 hits because there are 20 panels of 20 markers on the panel. But if you use sequencing, we have already seen that the average patient has about 5,000 changes. So we think that looking for 20 things is not so important in the future. And there is new data showing that many of these old school terminologies for high risk are not high risk. It really depends on how all the other 4,980 other markers are arranged. So I think most patients are told they have high risk, or not most, but many patients, they don't have high risk. And that's what we are seeing, and in particular, if we use the best drugs out there. And Ben can add to that.
I just want to add one quick thing for 10 seconds. So the FISH tests, we still use them. We're trying to move away from them. We already are, as Ola described. But I can give you one quick example. For example, Dr. Pandy in the back was looking at some genomic data earlier today even, and we were able to identify a case that was missed by FISH, but was able to be picked up by whole genome sequencing, an 1114 translocation that would have been missed. So these tests are good, but we can build on them, and we are.
Why do some patients have bone pain, other patients don't have no bone pain at all?
So around 90 or so %, if you look in big databases of patients that are newly diagnosed, have bone pain. And I think it's probably because myeloma is a disease of the bone marrow. It's a subset of the cells in the bone marrow, the plasma cells. When there are abnormal plasma cells, that's what the myeloma cells are. And they are located in the bone marrow. And the bone marrow is in the center of every bone in the body. The skeleton is made up by hard bone, and in the middle, soft bone marrow. So if there is a disease process in that marrow, the surrounding tissue, the bone, gets impacted in the majority, 90%. And that's usually where the pain comes from. When we treat the myeloma, when the myeloma cells disappear, the pain usually goes away.
Okay, well, I'm going to wrap things up. Please, these doctors are going to be here. So please come up to them and ask if you have a question that you haven't answered. And I'd like to thank the entire myeloma team, the development team, for putting this event on tonight. And I'd like to thank you all for being here and supporting us.